Sensor traces dopamine released by single cells. New technology could help neuroscientists understand how dopamine influences brain activity. MIT chemical engineers have developed an extremely sensitive detector that can track single-cell secretion of dopamine, a brain chemical responsible for carrying messages involved in reward-motivated behavior, learning, and memory. Using arrays of up to 20,000 tiny sensors, the researchers can monitor dopamine secretion of single neurons, allowing them to explore critical questions about dopamine dynamics. Until now, that has been very difficult to do. Now, in real time, and with good spatial resolution, we can see exactly where dopamine is being released, says Michael Strano, the Carbon PDubs professor of chemical engineering and the senior author of a paper describing the research, which appears in the Proceedings of the National Academy of Sciences the week of February 6. Strano and his colleagues have already demonstrated that dopamine release occurs differently than scientists expected in a type of neural progenitor cell, helping to shed light on how dopamine may exert its effects in the brain. The paper's lead author is Sebastian Kruss, a former MIT postdoc who is now at Göttingen University, in Germany. Other authors are Daniel Salem and Barbara Lima, both MIT graduate students, Edward Boyden, an associate professor of biological engineering and brain and cognitive sciences, as well as a member of the MIT Media Lab and the McGovern Institute for Brain Research, Lila Vukovic, an assistant professor of chemistry at the University of Texas at El Paso, and Emma van der End, a graduate student at Northwestern University. A global effect. Dopamine is a neurotransmitter that plays important roles in learning, memory, and feelings of reward, which reinforce positive experiences. Neurotransmitters allow neurons to relay messages to nearby neurons through connections known as synapses. However, unlike most other neurotransmitters, dopamine can exert its effects beyond the synapse. Not all dopamine released into a synapse is taken up by the target cell, allowing some of the chemical to diffuse away and affect other nearby cells. It has a local effect, which controls the signaling through the neurons, but also it has a global effect, Strano says. If dopamine is in the region, it influences all the neurons nearby. Tracking this dopamine diffusion in the brain has proven difficult. Neuroscientists have tried using electrodes that are specialized to detect dopamine, but even using the smallest electrodes available, they can place only about 20 near any given cell. We're at the infancy of really understanding how these packets of chemicals move and their directionality, says Strano, who decided to take a different approach. Strano's lab has previously developed sensors made from arrays of carbon nanotubes, hollow, nanometer-thick cylinders made of carbon, which naturally fluoresce when exposed to laser light. By wrapping these tubes in different proteins or DNA strands, scientists can customize them to bind to different types of molecules. The carbon nanotube sensors used in this study are coated with a DNA sequence that makes the sensors interact with dopamine. When dopamine binds to the carbon nanotubes, they fluoresce more brightly, allowing the researchers to see exactly where the dopamine was released. The researchers deposited more than 20,000 of these nanotubes on a glass slide, creating an array that detects any dopamine secreted by a cell placed on the slide. Dopamine diffusion. In the NUPNAS study, the researchers used these dopamine sensors to explore a long-standing question about dopamine release in the brain: from which part of the cell is dopamine secreted? To help answer that question, the researchers placed individual neural progenitor cells known as PC12 cells onto the sensor arrays. PC12 cells, which develop into neuron-like cells under the right conditions, have a starfish-like shape with several protrusions that resemble axons, which form synapses with other cells. After stimulating the cells to release dopamine, the researchers found that certain dopamine sensors near the cells lit up immediately, while those farther away turned on later as the dopamine diffused away. Tracking those patterns over many seconds allowed the researchers to trace how dopamine spreads away from the cells. Strano says one might expect to see that most of the dopamine would be released from the tips of the arms extending out from the cells. However, the researchers found that in fact more dopamine came from the sides of the arms. "We have falsified the notion that dopamine should only be released at these regions that will eventually become the synapses," Strano says. "This observation is counterintuitive, and it's a new piece of information you can only obtain with a nanosensor array like this one." The team also showed that most of the dopamine traveled away from the cell, through protrusions extending in opposite directions. Even though dopamine is not necessarily being released only at the tip of these protrusions, the direction of release is associated with them, Salem says. Other questions that could be explored using these sensors include how dopamine release is affected by the direction of input to the cell, and how the presence of nearby cells influences each cell's dopamine release. The research was funded by the National Science Foundation, the National Institutes of Health, a University of Illinois Center for the Physics of Living Cells postdoctoral fellowship, the German Research Foundation, and a Liebig Fellowship.
When you back it up and you drop it down low.